Radioactive labeling is a powerful technique in molecular biology, biochemistry, and genetics that offers several advantages for tracking and detecting biomolecules. However, it also comes with some disadvantages. Let's explore both the pros and cons of using radioactive labeling. Advantages of using radioactive labeling. 1. High sensitivity. Radioactive labeling can detect very small amounts of nucleic acids or proteins, often down to picomolar levels. This sensitivity is particularly helpful in studying gene expression and binding assays. 2. Quantitative. Radioactive signals can be accurately measured using instruments like scintillation counters, enabling precise quantification of biomolecules in a sample. 3. Stable and uniform labeling. Isotopes like superscript 3, superscript 2p, and superscript 3s are incorporated evenly into nucleic acids and proteins without affecting their biological activity. 4. Versatile for many assays. It's widely used in blotting techniques, hybridization, enzyme assays, and binding studies. 5. Reliable detection. It's particularly useful for studying biomolecule interactions, such as protein DNA binding or receptor-ligand interactions. Disadvantages of using radioactive labeling. 1. Health and safety concerns. Handling radioactive materials requires strict safety protocols to minimize radiation exposure, increasing the complexity and cost of experiments. 2. Environmental and regulatory issues. Proper disposal of radioactive waste is regulated and costly, posing environmental hazards if not managed correctly. 3. Limited shelf life. Radioactive isotopes decay over time. For example, superscript 3 superscript 2p has a half-life of 14.3 days, meaning researchers must time their experiments carefully. 4. Labor-intensive. Working with radioactive materials requires specialized equipment, additional training, and careful handling. 5. Non-radioactive alternatives. Safer alternatives, like fluorescent or chemiluminescent labeling, offer nearly the same sensitivity without the risks of radiation. 6. Storage complexity. Radioactive materials need special storage and handling to comply with regulations and avoid contamination. Summary of pros and cons. Advantages include high sensitivity, quantification, and versatility, while disadvantages involve safety risks, regulatory concerns, limited isotope shelf life, and the rise of non-radioactive alternatives. Conclusion. Radioactive labeling remains a sensitive and reliable method for studying nucleic acids and proteins, but its usage is declining due to safety concerns and the availability of non-radioactive alternatives. Researchers must balance the need for sensitivity with the practicality of handling radioactive materials.